All right, hello everyone. Today is Tuesday, February 16th, and we are welcoming you to the OSU Specialty Crops in Ohio Controlled Environment Ag Center Virtual High Tunnel School. This school is a live weekly seminar and discussion for current and potential high tunnel users. And classes started January 12th and will run through today, February 16th, being that today is session number 6. So thank you for joining us. We recognize that there's a lot of competition for your time, so we appreciate your sharing some of it with us. I'm Haley Shoemaker, and I'm a program coordinator with Ohio State University Extension, and I'll serve as your host and moderator for today's session. We will welcome and hear from our presenters soon. However, before getting into the subject matter, we do need to cover a little bit of background and housekeeping to help you get the most from joining us today. First, please note that you currently cannot be seen or heard, and that's okay. Your audio and video transmissions are currently blocked, and I'm currently the only one that can be seen and heard. Second, your video will remain off throughout the class, but you do have 2 options for communicating with us, the presenters, and other attendees during class. The first is to use the Q&A feature available by clicking Q&A when you hover over the bottom of your Zoom screen. This will open a box allowing you to type a question or comment and direct it towards everyone or someone you select. The second way to communicate is by using audio. To use that option, wait until the Q&A portion of the session and then click Raise Your Hand at the bottom of the window. I will see that you've raised your hand, and if possible, I'll ask you to unmute yourself and speak. Please note that placing a question in the chat box or the Q&A is very effective, and if the question is not answered today, it will be addressed after class. All registrants will receive a follow-up email after the class today containing responses to questions and comments that were placed in Q&A. So now that covers how to communicate with us during the session, let's get started. Today, we're going to cover soil biology, chemistry, and tilth. Are they holding you back? And we have 2 guests that you're sure to enjoy hearing from on that overall topic. Joining us today are Matt Kleinhenz and Anna Testen, and Matt will start by speaking on maintaining soil health and the challenges and solutions that are associated with that topic. And then following Matt will be Anna, who will be presenting on soil-borne diseases and what we can do about them in high tunnels. Matt, all that being said, let me turn the floor over to you. And what should we know about maintaining soil health?
Okay, great. Thank you so much, Haley. I hope that everyone can see the screen with a red background and the title slide. That's true. We can get right into the topic at hand. What I'd really like to emphasize in the next 15 minutes or so is what you're seeing here on the title slide, which is that maintaining soil health in the high tunnel is one of the keys to your long-term success in high tunnel production. Those of you who are somewhat new to high tunnel production will come to discover this, and I think that some of you who are experienced high tunnel users have already discovered it, and so this presentation will be a bit of a refresher, reminder, and prompts to consider other items. And for those who are less experienced with high tunnel production, it might be a bit of a list of items to take into consideration going forward. Throughout this school, we have attempted to provide a balanced perspective on high tunnel use and season extension, providing what I think is an overview of both its pros and cons. And certainly we're very familiar with the benefits of high tunnel production, but we also need to be aware of some of its associated challenges. And it's my understanding, along with others in the research extension and farmer communities, that the deterioration of soil health or soil quality or the productivity of that soil is among the challenges associated with high tunnel production. So that's the focus of today. And I think the reason for that is that we expect a lot of our soils. We put a really heavy burden on our soils in high tunnel production often. And these are some of the specific burdens that we do place on our soil. I won't go through them each in detail, but as you can see the array of standard practices before you, you will appreciate that you might be familiar with each and every one of them or most of them. We often are growing crops with short rotations with small and shallow root systems. We're not returning very much residue to the soil. The high tunnel environment is very unique. It's been described as an irrigated desert. So these are some of the risks that we place on our high tunnel soils, or burdens, if you will, that we place on high tunnel soils. And the effects of those individual burdens can be more than additive. It can be more than just 2 + 3 = 5. It can actually be 2 × 3 = 6, multiplicative, if you will. And all of them coming together have the potential to reduce your costs, increase your costs, lower your productivity, and lower your profit potential, for sure.
Hey Matt, not to interrupt, but you're like cutting in and out. You sound real good, and then you're real soft. So I don't know if it's.
I will attempt to speak up. I don't know if it. I mean, I'm coming
Matt,
through.
it looks
Yes.
like when you're leaning towards the computer that your audio is picking up your voice better.
Okay, I'll sit closer. Thank you.
Thanks, Matt.
I'll do my best. So the specific soil health, soil quality, soil productivity issues that I am familiar with in high tunnel production are listed here. Reduction in soil structure, reduction in organic matter, increases in compaction, salinity, nutrient imbalances, and disease inoculum. These are just a number of the soil health related issues in high tunnel production with which I am familiar. And the research, as I say, the research extension and farming community is also aware of these and other challenges. And so, a number of steps have been taken to survey the farmer community and its familiarity with soil health challenges in high tunnel production. And that work is ongoing. I'm just one person, and I'm providing this slide as one bit of evidence that those three communities are taking up the question of how to manage high tunnel soil health on the long term most effectively. So just as one person, these are the places and over the time period that I have been asked to address the overall topic of soil health and high tunnel production as well. Most of us would agree that if you commit to driving posts into the ground, putting a roof arches over the top of them, establishing a high tunnel or more high tunnel, covering, of course, the frame with plastic, taking all the normal steps that you do in installing a high tunnel, and then beginning to crop it, probably in the summertime at minimum, but perhaps in the fall, winter, and spring as well. Dealing with all the things that you can end up having to deal with in your high tunnel-based business, including some of the circumstances that some of us are experiencing here recently. If you make all those commitments, it could be said that one of the largest commitments that you are making is to the soil beneath the high tunnel, the soil that is being covered by that high tunnel. And so the purpose of today and ongoing communication would be to help ensure that that soil resource is as effective for you as it possibly can be for as long as it can be and as needs to be. And what we know about soil health, in my view, based on research done in open fields, open sky production, dwarfs what we know about the health of soils in high tunnels. There's just much more known about open field farming, open sky farming, than there is about high tunnel farming. But there's a lot of research underway, and so I encourage you to stay tuned in that area. Most of us would agree that it's easier to lose soil health than to maintain it at some level. It's something that goes away quite easily. And it's not maintained by accident. And one needs to take specific, conscious steps and make specific conscious decisions to maintain the health of the soil in the tunnel. We could discuss at length what soil health is. We know that it's a combination of physical, chemical, and biological attributes. They overlap, and each of them have their own individual characteristics or examples, for sure. There are tools and approaches for assessing soil health, widely available to you for your own reading and application. They'll steer you towards looking at various parameters of your soil or engaging a research extension team and doing the same. Very sophisticated instruments, tools, kits, resources for you to assess the health of the soil in your high tunnel. But sometimes it's a matter of just stopping and picking some up, but doing so very, very purposefully, knowing where you're collecting it from and knowing what you're going to do with it once it's in your hand. Do you use a soil probe? Do you use stratified sampling? Do you separate the sample from 0 to 2 inches, from the sample from 2 to 4 inches, from the sample from 6 to 10 inches when you submit that for analysis? Because the parameters in that soil, the characteristics of that soil differ from top to bottom. And so to grab a whole core that goes from 0 to 8 or more inches and lump it all together, you get an average, but that's not necessarily what your roots and your crops experience. When you soil test, what philosophy is the lab using when they make recommendations? Do you account for the nutrients that are removed by the crop? What are the optimal levels for high tunnel production relative to field production, for example? Lots of important questions to be addressed when developing a crop management plan specific to high tunnels and specific to keeping the soil in good health. Other indicators can be measured in the field. You can look at penetration resistance, water infiltration rate. It's as simple as putting some marks on a spade, leaning on that spade with a known force, and just seeing how far the spade will go in. I'm sure many of you are familiar with this practice. You can use a fancy gadget to actually get a number that provides you with that resistance, but that spade test will also partially suffice. You can cover a certain small area of soil with plastic, pour a known volume of water in it, pull the plastic out, start the stopwatch, and see how long it takes the water to drain inward, to clear the top of the soil as a measure of infiltration rate. Regardless of the steps that you take, these are some of the challenges that you're likely to be addressing or experiencing, perhaps. The approaches that you're likely to employ in overcoming the challenges I mentioned earlier. There's a list of them here. We're going to hear a lot about ASD, so I want to leave plenty of time for that. So I'm just going to focus on the ones that follow ASD. You'll hear from Anna on ASD. She might reference these pictures like these or other fact sheets, and she's already mentioned fact sheets in the chat box. I'm here to mention rotation and amendment as two approaches to maintaining the health of the soil in the tunnel. But also, as you amend the soil, as you apply things to it, do a robust job, a very solid job of analyzing the amendments that you're applying. And if possible, rotate those amendments. Repetitive, frequent, long-term applications of the same material to that soil will drive it in a direction likely to result in nutrient imbalances or excesses. Likewise, an overhead sprinkler system may help with your crop rotation. We have released a guide on how to install an overhead irrigation system in a high tunnel if needed. This can provide assistance when you're trying to establish a crop that you would otherwise be more difficult with drip irrigation, which is very, very common in high tunnels. So if you're interested in that process, it's a relatively straightforward one. If you're able to remove the cover and let the wind and the rain and the snow help purge those same salts, that's also a very, very effective step. The freezing and thawing action of soil mixes soil as it expands and contracts due to the freezing and thawing action. It actually mixes. That can actually rotate the layers top to bottom, help with that process. And in some cases, you might want to pay very, very close attention to your irrigation water. Have it tested if you have not done so already, because it could actually be the source of some of the unwanted chemistry that's taking place in chemical changes taking place in your soil. Have you asked the question if deep tillage might be useful to you? This is a common practice in some field-based production, but it's less often used in high tunnel production, to my knowledge. But it might be a tactic that will benefit some people if they're able to use, say, a deep broad fork or other implements to help fracture that soil, maybe the plow pan or the hard pan that's beneath the upper layers. And ordinarily, we, as you've heard me say and Brad and others say, we want to avoid flooding inside the tunnel at all costs. We want the only water in the tunnel to be water that you put there on purpose and probably for irrigation. But there is a time, maybe a time for you that flooding the tunnel on purpose can help purge the excess salts that are likely to build up due to fertigation, fertility practices, and other circumstances. So keep that option in mind as well. If you're unable to purge, if you're unable to use a coverless fallow, take some of the other steps, consider a movable high tunnel. They do have advantages. If you're unable to reclimate, so to speak, and maintain the health of the soil beneath the tunnel, for some people, moving the tunnel is appropriate. But for that, of course, requires a specific design to make that possible. And then once that soil is uncovered, we're talking about the rain, snow, precipitation, normal farming practices and environmental conditions that would help reclaim, if you will, and maintain the health of that soil. This high tunnel is easily moved. You can watch videos of the process, and it has shown for us that very clear opportunity to use soil management practices that are consistent with open field production, yet still get the benefit of a high tunnel from time to time. So again, a fairly straightforward process, and a movable tunnel is a partial solution for some people, no question. And I'm showing you pictures here of the locations and of the types of manufacturers, examples of the manufacturers that offer movable tunnels. For some, container production is the answer. They outright will say, I'd rather bring in my medium, my rooting medium. I would rather not rely on the soil that my high tunnel covers alone. I would rather use container-based production underneath my high tunnel. That is a completely legitimate step to take. Is simply one to be taken with due caution and due respect for the costs and the associated changes in the practices that accompany it, as you will. But container-based production under a high tunnel or within a high tunnel is quite reasonable in terms of a strategy to helping to mitigate the soil health related challenges associated with high tunnel production. So the partial solutions are some of the ones I've listed here. One final one that I'll just toss out to you for consideration is the question of the relative merits of having few large tunnels or more smaller tunnels. What role would that have in maintaining the health of the soil in each of the tunnels? What impacts would it have on your farm business to have more smaller tunnels versus a few larger ones? That's just something that we can discuss at another time. So in summary, and to make room for us to hear from Anna and address a few questions, I'm just encouraging you that regardless of the approach that you've taken to your soil health and quality management, remember that you've paid for the opportunity to include high tunnels in your business. You would be prudent for you to recognize that soil health is key to maintaining that high tunnel as an asset. Various strategies are available to you to help maintain the health of the soil in the high tunnel, and that even small investments in that process will matter. They will offer you a return on investment. So Haley, I'll pause here. We can address any questions if there is time for them. Otherwise, we can hear from Anna and then regroup and have other questions as well.
All right, thank you, Matt. Just a couple questions in the chat box right now as Anna gets her presentation pulled up. The first is there a link to the document mentioned about installing an overhead irrigation system in a high tunnel?
Currently, there is no link. It's literally brand new as of 2 days ago in terms of finalizing the document, but I can easily provide one to all the registrants, or you can email me separately and I can email you the document. But the link will be available at some point.
Okay, thank you.
Hey, Haley, can you let me share the screen, please?
Absolutely.
Thank you.
I apologize for the audio problems. They are unpredictable on my end. Difficult for me to troubleshoot because everything checks out when I do my test before the presentation, before the session. So I'm not sure where the where the challenges exist exactly. But I hope it came through well enough. Are there any other questions, or should we move on?
We did have one other question while I get Anna plugged over here. Other question was, in all the years OSU has used high tunnels, do we have any data on the best ways to maintain soil quality?
There have been for quite a few years that OSU has been involved with high tunnel use, and I think the best steps, if I'm hear the question correctly, the best steps are the ones encapsulated in the last 15 minutes or so. It's not one strategy; it's multiple ones, and it will vary slightly based on each person's operation, including whether or not they're farming organically or not. So this is not a silver bullet kind of scenario. This is more of a scenario in which one would apply multiple approaches to ensure that their soil remains productive for them, and that's going to involve crop rotation, crop selection and rotation, this careful selection of amendments, including fertilizers, the recognition that it's not raining and snowing inside the tunnel, and you're not benefiting from those normal occurrences that you are in field-based production. You may be using narrow rotations, meaning very few crops. So it comes with a bit of recognition and then a bit of application of multiple strategies to address the problems that may or are likely to arise. And I would say, as a final word on that before we transition, testing is essential. Testing is essential. And for help on that, you can reach out to anybody within Extension or myself for guidance on that point. But even what we call a simple soil test needs to be approached, I think, very, very carefully and somewhat differently in the high tunnel than in the open field scenario, beginning with understanding the different levels, layers in the soil. It's like a stack of pancakes, if you will. There's a top, a middle, and a bottom, and the characteristics of those layers may differ. So being mindful of that when you sample is important.
Okay, and I am going to, Matt, while I pull up this next question, since you are the host, can you try and give Anna the allowance to share her screen? There's some technical difficulties on my end.
Yeah, there should. Oh, panelists. There we go. It should be. Anna, you should have it now.
Okay, there we go.
Great.
All right.
Matt, you do have a few more questions, but we can put those towards after Anna's presentation.
Okay. Yeah, perfect.
So Anna, we're going to turn the floor over to you. You're going to talk to us about soil-borne diseases. So we're looking forward to hearing your topic.
All right. Can everybody see my presentation?
We can see the full screen.
Hold on one
your
second.
PowerPoint.
All right. How's that looking?
Perfect.
All right. All right. Well, good afternoon. Good morning, everybody. My name is Anna Testen. I'm a research plant pathologist with USDA ARS. And today I'm going to share with you some research that I started 5 years ago when I was a PhD student in Sally Miller's lab. And this is something that I think is really important to consider when you're considering the long-term sustainability of your high tunnels, and that's soil-borne diseases. And many times growers that we're working with don't notice these diseases until it's too late. So let's get my computer woken. Come on, let's go to the next screen. All right, so why should you think about soil-borne diseases as a special issue in your high tunnels? First, I want everybody to take a look at this tomato root. So this is what you should think of when you're thinking of a healthy tomato root system. But oftentimes when I go into a high tunnel, this is not what I see. Because in high tunnels, we have these special protected microenvironments. Within these high tunnels, we have few rotation options. The soil-borne pathogens that we're dealing with have an extremely wide host range. And so regardless of what you're rotating, you're always providing food to these soil-borne pathogens. And again, in these high tunnels, we have warmer environments that have fewer temperature extremes. So we don't get these winter kills. And so if you have soil-borne pathogens, you have less reduction in these pathogen populations. So what happens is you have the perfect conditions for the buildup of these soil-borne pathogens over time. Now, what should you look for in your high tunnel that would indicate that you had these pathogens? Now, this is a picture of an extreme case. This is a high tunnel in Southern Ohio in which the grower had been growing tomatoes for 15 years. So, this is a really good picture though, because it shows what you should look for. So the two things you're going to see as soil-borne diseases start in your high tunnel is you're going to notice that your plants are stunted and yellowed. Those are your first indication that those plants are starting to be stressed due to some root issues. As soil-borne diseases and their pathogens build in soil, you're likely going to see more severe symptoms, including wilting and dieback. And if you see this, this really should clue you in that you probably have an issue in your soil. Now, this is what to look at above ground, but everybody should take and make it part of their routine to start looking at their roots and crowns at the end of the season. So what you should look for on roots, crowns, and stems in terms of soil-borne diseases include girdling or browning at the soil line. That would indicate that you have something that's affecting those crowns. You also want to look for internal vascular discoloration, and I have some slides later that show this better. That could indicate that you have some sort of fungal wilts. Looking at root health in general, if you have reduced root mass or a lack of fine roots, that indicates that you have some sort of soil-borne pathogen nibbling away at the root structure. You also want to look for discoloration of the roots, and that can range from brown, gold, or black discoloration. Sometimes unhealthy roots are soft and fall apart easily. This is a good indication that you have an oomycete problem in your soils, like Pythium or Phytophthora. And then also roots can be deformed when you have soil-borne issues. This can include a barkiness of the roots, or it can be a galling or club-like growth. And if you take a look at this picture over here, I want everybody to understand this is what a really unhealthy tomato root system looks like. So you can see this brown discoloration on these roots, and you can see this barkiness and this galling. So this root system is just loaded with soil-borne pathogens. And this actually was taken from that high tunnel that I showed you in the previous slide. So what diseases are we talking about specifically? So there's a range of diseases that can occur in the soil in high tunnels. Now, these diseases can occur alone, but unfortunately, these diseases occur more often in complexes of one or more of these pathogens. So we're dealing with fungal wilts, fungal root rots, damping off, white mold, and root knot nematodes. So that's a lot to deal with. So let's go into now a little bit better descriptions of these individual diseases and what to look for to see if you have these as an issue in your high tunnel. For Verticillium wilt, this is a pathogen with a really broad host range. What you want to look for in tomato are these really distinctive V-shaped lesions on the leaves. They're very characteristic of Verticillium wilt. For any sort of fungal wilt, either Verticillium or Fusarium, as I said earlier, you can look for vascular discoloration. So you can either take a knife and cut across that stem or along that stem. What you want to look for is this brown discoloration of your vascular tissue. So there you're seeing damage from that fungus blocking the vessels in the plant. So as I said earlier, Verticillium has this really broad host range. And this can be a clue, you know, if you're rotating different crops in your high tunnels and you have issues with wilts every year, that's a good indication that you might have a Verticillium issue. So here's a high tunnel with strawberries in it, and you can see that these lower leaves are wilting. And in fact, this grower was rotating with peppers and strawberries and had issues with Verticillium in both of his crops. Now, Fusarium wilt is another issue in high tunnels. In tomatoes, this has a really characteristic symptoms that the yellowing and dieback only occurs on half of the plant. And you can see that very clearly, only one half of this tomato has leaves that are yellowing and wilting. Now, a new disease that's a major issue in Ohio high tunnels and likely in high tunnels in surrounding states is corky root rot caused by Pyrenochaeta persicae. Now, this is a really important disease to look at the end of the season in your tomatoes, and it causes very distinctive symptoms. You see this tree-like bark and cracking on your roots, and this brown cracking usually occurs in characteristic bands. So you have healthy root, brown cracking, and a healthy root again. So these are a good indication that you have corky root rot. Black dot root rot caused by Colatotrichum cocodes is another soil-borne issue in high tunnels. And this one is interesting because most people know this as anthracnose on the tomato fruit. So this is a disease that can affect both your fruits and your roots. Now on roots, this causes a very distinctive honey brown discoloration. And if you take a hand lens or a magnifying glass, you can look at that more closely and see these black speckles on the roots. Root knot nematodes are also another emerging issue in high tunnels. And this is mainly because these high tunnels, again, are warmer and they're more protected from the winter. But what you want to keep an eye out in terms of nematodes is you're going to look for this galling or this bead-like deformation of your roots. And that's a very good indication, regardless of crop, that you're dealing with root knot nematodes. Something else you can see if you have a hand lens or again, a magnifying glass is you can see these brown or golden root masses or egg masses of the nematodes on those galls. So 3 more diseases to look out for in your high tunnels include Rhizoctonia. This has a very characteristic symptom of girdling. So this is girdling on a cabbage plant. And you also get this dry brown root rot. So these roots with Rhizoctonia root rot are not normally soft. When you're looking at Pythium and Phytophthora root rot, now these are root rots that are usually going to be soft or wet. And you're going to have a brown or golden discoloration. And these are caused by oomycetes, so they really thrive in wet, compacted soils. Now, the last disease, which I'm sure everybody has seen in their high tunnel at some point, is timber rot or white mold caused by Sclerotinia Sclerosiorum. So for this, you're going to look for bleaching of the stem. That stem is going to be hollow, and inside that bleached area, you're going to see some white fungal growth, and also sometimes you'll see some large black sclerotia or survival structures. Now, Sclerotinia is tricky because if those sclerotia fall into the soil, it can be soil-borne. But because this has such a broad host range, the spores of this fungus can also blow into your high tunnel as well. So it can be tricky to identify whether it's a soil-borne issue in your high tunnel or it's coming in from outside. So, okay, so that was a really quick introduction to how to identify these diseases. But what's more important is to think about how we manage soil-borne diseases, because management of soil-borne diseases is a bit different from management of foliar diseases. And it's because once you have these soil-borne pathogens in your soil, they're there for quite a while and can be difficult to manage. So when we're thinking about managing soil-borne diseases, we want to do 2 things. We want to prevent existing pathogen populations from increasing. We don't want a bigger problem than we already have. So we want to keep disease pressure low. And to do this, we use management practices that promote general plant and soil health. And we also use management practices that prevent pathogens from multiplying, such as using a resistant variety, grafting on disease-resistant rootstocks, or rotation. Now, at the same time, we want to reduce those existing pathogen populations. So again, we don't want to have a bigger problem. And this usually involves some form of soil disinfestation. So let's look at what our core practices of soil-borne disease management are in high tunnels. First, you always want to think about adding organic matter. As Matt said in his talk, these are closed systems. They don't have those inputs you have in the field. So think about adding compost or some sort of cover crop-based organic matter into your high tunnel. You want to deal with any soil health issues such as compaction or poor drainage, because those really promote the formation of unhealthy roots. Sanitation, as in any disease management, is very important. So make sure at the end of the season, you're removing as much root material as possible. In terms of diseases like black dot root rot, you want to make sure you're removing those culled fruits. And then for any plant that has white mold, you want to remove that from your high tunnel as soon as possible because you don't want those sclerotia falling into your soil. Rotation is very important, and you'll see on my next slide, this can be difficult in high tunnels because of the limited number of crops we have available to us that have resistance. But at the very least, make sure you're rotating out of families. So don't follow tomato with pepper or eggplant, but think about going from tomato to maybe something like strawberry or brassicaceous disease. And then whenever possible, if this fits into your production practices, make sure you're selecting resistant varieties. And there are good options available to growers for that have resistance to Fusarium and Verticillium. So this is a graphic I put together showing the susceptibility of these different high tunnel crops to soil-borne diseases. So on the top, we have our different crops, and down the right-hand side, we have the different diseases that occur commonly in high tunnels. Now you can see that for poor tomato, it's susceptible to all these different diseases. And luckily tomato will keep me employed for the rest of my life. But if you have issues with a disease like corky root rot in your high tunnel and you don't want to graft or use some other management option, you can consider moving into lettuce, brassicaceous crop, or even strawberries, which are not susceptible to these diseases. Now, if you have an issue with Verticillium wilt, you could consider something like a brassicaceous crop that it has a lower susceptibility to this pathogen. Now for Fusarium wilt, Fusarium is interesting because it is a little more host specific. So if you have Fusarium wilt in cucumbers, it likely is not going to affect your peppers and tomatoes. So think about this as you're designing your rotations. So when we have to take a more active management approach to reduce soil pathogen populations, as I said, we have to use some sort of disinfestation of our soils. There has been some research showing that use of cover crops and biocontrol can reduce pathogens in soil. But as Matt said earlier, a lot of this is open field based and we don't have good evidence for high tunnels. So this research is ongoing. Again, make sure you're selecting resistant varieties. Now, if resistant varieties is not a tool for you or you really have issues in your high tunnels, consider grafting onto disease-resistant rootstocks. This is really important disease management tactic, especially for tomatoes, and is really good for managing corky root rot in high tunnels. So steam sterilization and solarization are two options that you can use in high tunnels, but in a more temperate Midwestern setting, they can be difficult. It can be hard to get the equipment in for steam sterilization, and for solarization, we usually don't get temperatures in the Midwest for long enough periods of time to reduce these pathogen populations. So my favorite soil-borne disease management tactic is anaerobic soil disinfestation. I'll talk about that in the next few slides. And I do want to make sure that everybody today knows that fumigation is not a viable option for reducing soil-borne diseases in high tunnels. Most fumigants are not labeled for high tunnel use, and they're just not a practical option for growers. So anaerobic soil disinfestation, or ASD, is a three-step disease management process in which you amend soil with a carbon source, such as wheat bran or molasses. You then work that carbon source 6 to 8 inches into the soil. Then you irrigate that soil to saturation to fill all the pores in the soil with water. And then you tarp that soil with plastic mulch for a period of 4 to 6 weeks. So what happens in this time is that microbes that live in the soil break down this added carbon source and they use up all the oxygen in the soil. And what happens when they use up all the oxygen and break down this carbon source is they produce a lot of byproducts that are toxic to soil-borne pathogens. So it's a really interesting natural way to manage diseases. Now, we've had good success, again, starting back in 2015 with trials in high tunnels across Ohio, that we see really good efficacy of this treatment for reducing soil-borne diseases, including root knot nematodes, which seems to be the most sensitive to ASD, Verticillium wilt, and corky and black dot root rot. So there's other research from the Miller lab that also showed reduction in Sclerotinia, but that was not in a high tunnel system. So just to show how efficacious this is an example from one of our trials. In our control plots, we had about a 60% root rot, and following ASD, that root rot went down to less than 15%. So that's really great to see that impact of the treatment. So for applying ASD, because we don't need super high temperatures, it really fits into rotations a lot better than a tactic such as solarization. So we recommend that growers apply ASD in either September or in the first week of October. And in Ohio, if you have decent temperatures in your high tunnels, say at least above 70 degrees, 4 weeks of tarping is sufficient. For a carbon source, either wheat bran or wheat midds at 9 tons per acre, so you do apply at a pretty high rate, is effective, or a combination of wheat bran or midds plus molasses. Now, as with any disease management practice, we want to combine this with grafting and other management techniques. So that wraps up what I wanted to talk about today. And if there's time for any questions, I'd be happy to take them. The links that are on this slide, I posted in the chat.
Awesome. Thank you, Anna, for a very informative session. So we do have a couple questions, and I think we have some that are more just general, but let me look and see if there's any specific. We do have a question. Are you ever adding biochar to improve structure and housing for the microbiology?
So in my work, I've never worked with biochar. I have seen some general research looking at soil-borne diseases, but not any research regarding biochar and the diseases specifically of high tunnel vegetables like corky root rot and Verticillium or Fusarium wilt.
Okay. The next question is, would any soil sterility be caused through any of these practices?
Yeah, so one of the interesting things about anaerobic soil disinfestation is that you're relying on the indigenous microbes in the soil. So following ASD, you do have shifts in your soil microbiome, but you don't have an elimination of the microbes in your soil. So usually what's been shown in other research is some of your beneficials will be knocked down following ASD, but following one cycle of cropping, they'll be right back up where they were before the treatment. But your pathogens, because they're a little less competitive, will still be knocked down.
Okay. And after ASD, what do you do to prepare the soil again so that's once again porous and aerobic?
Yeah, so that's a good question. So if you're doing raised beds in your high tunnel, you can treat those raised beds with ASD, and following treatment, you can just cut your planting holes into those soils, let it air out for a week, because following treatment, we do have some residual toxins and those soils smell terrible. So you let those air out and then you can plant as normal. Now, if you do ASD in your high tunnel, like as a flat treatment, you do your treatment, afterwards you'd pull off that soil and then just wait for it to be dry enough to work the soil again.
Okay, and the tarp that you're using, should it be permeable or non-permeable?
Yep. So you want non-permeable because you want to trap those gases and you don't want any air getting through that. So what some of our growers have used to save money is they've been able to use old covers from their high tunnels. And, you know, if there's any holes, duct tape is a good solution. You just tape over those holes. Otherwise, you know, you can get pretty wide, heavy gauge plastic from Lowe's or Home Depot for a reasonable price. There's been some research looking at silage tarping, and that also should be effective for soil cover.
And what other carbon sources would work for this, if any?
Yeah, so that's a great question. So we have some research where we were looking at broader options for carbon sources. So we found that soybean meal, wheat midds, sorry, I'm thinking of all the ones we looked at. Distillers dried grains, corn gluten meal. Those were all good options for reducing diseases, but for the cheapest, most economical carbon source you could get, it was either wheat bran or wheat midds.
We also have a question. Is there any control for white mold?
Yeah, white mold is tricky. So you can use sanitation. So again, always start with sanitation so you're not getting the buildup of that. There is biological control available for white mold. It's called Kleinhenz. That can be effective in a high tunnel. There are no resistant varieties that I know of for white mold, but Sally is typing an answer. So I'm sure she'll provide more info.
And is it best to do an anaerobic soil disinfection of the entire high tunnel or only on sections?
Yeah, that's also a really good question. So in the trials I've done, it's always been in sections just because growers we need those control areas to compare with. And it seems to work well. If you want to save money, you can just treat beds or raised beds. But it's probably easier to just treat the entire high tunnel. Then you don't have to worry about any spillover with those within row areas that you'd be not treating if you just treated beds.
Okay, thank you. And would you recommend the tarping process in a rotation as a preventative?
Yeah, that's also a good question. So based on what we've seen in high tunnels, and we did a survey of soil-borne diseases in high tunnels in Ohio, it seemed like most growers started seeing soil-borne diseases after 3 years. So, if you want to use it as a preventative method, I would recommend doing the treatment every 3 to 5 years. I would not recommend doing it 2 years in a row back to back. At that point, you're probably at too high of a level of soil-borne diseases to see good efficacy.
Okay. And then it looks like we have one more question here asking if you've heard of a carbon source called Monty's liquid carbon.
I have not heard of Monty's liquid carbon, but I'm assuming that's just a general soil additive type of thing. So for the carbon sources for ASD, usually we're looking at agricultural byproducts that you can buy at a feed mill because these are readily available. There's been some research into using cover crops, but usually with cover crops, you don't get as good of an efficacy because those are a little bit harder for the soil microbes to break down quickly.
Okay, and just one final request. We've had a request for you to bring up the slide with the links on it real quick. And as you're doing that,
I'm gonna pop those links into chat again because then actually people can copy them.
Okay.
So I'll also add that, and if they're in your presentation, which I think they are, this presentation is being recorded and the presentation will be at the YouTube channel. And it can, of course, be paused at that place. But yeah, putting them in the chat will also help. But we've had some questions to whether or not people can scroll in the chat.
Okay. It looks like Brad would like to answer one of these questions in the queue. And the question is there a need in Ohio for certain crops to be grown? And also, is there a certain crop that is grown and offered in overabundance? An example for sale at farmers markets or other?
And that seems like a more general, most of the growers in Ohio are growing tomatoes. Matt, you can talk about what you've seen growers growing in high tunnels as well.
Yeah, no, I can very happy to comment. I think at some level, if you'd ask most farmers, at some point, every crop is available in overabundance or in deficiency, depending on the market price. But high tunnels, as has been shown earlier, can accommodate a very large number of crops, excuse me, in most areas. I know that on the line, we have people from all over the country, so we can't take a strictly Ohio or even Great Lakes Midwest perspective on this. But high tunnels can accommodate a very large range of crops. It really comes down to, in my view, what crops are adapted climatically to a high tunnel environment. Secondly, what crops can be grown profitably on a relatively small parcel that high tunnels often represent. And doing that math will lead you to probably conclude that it's a lot of the fruiting vegetable crops that can be done, such as the solanaceous group, pepper, eggplant, tomato, some specialty cucurbits, lots of root crops, lots of leafy vegetables. But Brad, you might want to add something onto this.
Yeah, I think we discussed this at one of the sessions, but anything can be grown in a high tunnel. You just got to look at the economic feasibility of whether that is economically feasible for your own farm. Now, for my farm, growing radishes was only economically feasible to grow them in a high tunnel when we had our winter CSA. So there it comes down to the individual one-on-one, what fits your operation. But I think Matt will agree, the research been done around the world, we can grow just about anything in high tunnels. It's up to you as the individual grower or farmer to determine whether that is an economically feasible option for your particular operation. And every operation is going to be different. So, and people eat 365 days a year. So there's a demand for produce just about year round.
Thank you, guys. There is one other question here, and this may be more suited for Anna. If you defined a soil-borne disease problem in one of the areas of your soil, how far away do you need to garden to not be affected by it, or can some of the preventative measures you suggested be a remedy?
So that's a good question. So soil-borne diseases are pretty slow moving just because a lot of them are just spores sitting in the soil. But I'm not sure what you're talking about in terms of, sometimes you could go, 10 feet away and you'd be fine. If you're talking about raised beds, if you went from one raised bed to another raised bed, you should be fine. But, if you're thinking about moving a distance away, make sure you're practicing good sanitation. So make sure you're not transferring any of that soil from the previous area to your new area, because you're just going to be spreading diseases. Within a high tunnel, soils in high tunnels mix more than you think they do, just as you work the high tunnel. So I saw there was a question about, could you rotate in areas within a high tunnel? And you could. I just think it would be tricky to keep that soil contained.
Okay, thank you.
Anna, let me comment if I could real fast. Sorry, Haley.
No, you're good.
Everyone can appreciate how much foot traffic and equipment traffic there is often in most high tunnel operations. And to the extent that that soil is contaminated in area A, and you walk through area A and walk into area B, the inoculum's traveling with you. Same way with the implements that you use. You use a spade over here, it's covered, got a little soil dust on it. You put the spade in the ground over there. You're inoculating your soil as you go. I mean, I'm not a pathologist, and Anna and Sally and others know this. But the holistic approach that Anna, I think, is advocating is the correct one. Understand the biology of the organism that you're dealing with, what it needs to survive in terms of a host and other parameters, and then ask yourself, where can you mess up the system for it? Where can you intervene? Where can you disrupt it? And it might go back to say, hey, buy high quality seed and don't plant the disease, exclude it whenever possible. And then, of course, when needed, use some other interventions like an actual spray or some sort of, crop protectant. But these are what we call, for those who are somewhat new to the process, what we call integrated pest management or integrated crop management approaches, all designed to, in a disease case, either remove the pathogen or exclude the pathogen, prevent the environment from being conducive to its growth, or don't give it a susceptible host. One or more of those three.
Thank you, Matt. And it looks like the other questions in the queue have already been answered or are in the process of being answered. If you have any other questions, please feel free to raise your hand or type them into the Q&A. Okay, we did just get another question that came in. Are there any positive nematodes or other things that can help build a healthy soil system against these issues?
So there are parasitic nematodes, but they're not going to be, unfortunately. So they are important components of a healthy soil, but you're not going to be able to take a bucket of parasitic nematodes and dump it into your soil and see a good effect. Unfortunately, it'd be awesome if we could do that, but we can't.
Yeah, this reminds me of because of the question of positive nematodes or other things. Certainly, various types of amendments, composts, some composts might classify it as something that can be applied to the soil to help build that system. The jury is still out as to whether or not what we call micro-containing biostimulants can do the same thing, or other biologicals. The jury is out on some of those, but stay tuned because more research is being done on them all the time. There are, at least from my perspective, some potential for targeted soil additives to be effective in specific cases, but you just want to proceed cautiously and based on information.
And it looks like we have just a couple more questions here as we wind down. The molasses role is to support the anaerobic process or something else. Is the sugar important part? Is the sugar an important part of that?
Yeah, so the carbon source, either whatever you choose, and it can be molasses, is you just need to provide food to those microbes. So one thing that most people are more familiar with of like overfeeding that makes things go anaerobic is when you get anaerobic areas in lakes. Too much fertilizer goes into that lake, algae blooms, and it uses up all the oxygen. Essentially, we're trying to do the same thing in soil. We put a ton of food into the soil, either wheat bran, molasses. Those microbes go crazy. They break it down. They produce a lot of nasty byproducts, and they kill the pathogens. And the molasses is one way to do that. Molasses is too much sugar. So usually you see better efficacy if you mix molasses with something like wheat bran. And somebody also asked about organic compliant carbon sources. So ASD does work in organic production, but Sally and I had discussions with OFA when this started. You just need to talk with your certifier about the carbon source that you want to use, if that's allowable to them to maintain your organic status. If they say your carbon source is not allowable, you can purchase organic molasses.
And kind of still on the molasses train of thought, is it granular or a diluted liquid?
Yep, so we do diluted molasses. And if you look at that fact sheet that we link to, it explains, molasses is tricky to work with. So normally we dilute it 1 to 4 and then pour it over the soil. Again, I would suggest working with something like wheat bran and add molasses kind of as a adjuvant, just something extra to add. It is tricky. I see following up, somebody asked about the application rate. So again, that's in the fact sheet. So for your wheat bran, you want 9 tons per acre. And molasses, you add at a lower rate of 4.5 tons per acre. The molasses, you could add at a low rate of like 1 ton per acre. When you have the wheat bran there, the molasses is just kind of like lighter fluid. The microbes will start using that molasses up quickly, and then they'll start breaking down the wheat bran.
Thank you. And in general, how many years of a non-host plant can degrade the soil disease pathogen?
Oh, that's a good question. One tricky thing with these soil-borne pathogens is their survival structures can survive many, many years. So some of these can survive in the soil for 10 plus years. So, you're not going to eliminate everything with rotation, but you will knock down those pathogen populations. So usually like a 2 to 3 year rotation will knock down some of these pathogen populations. But again, in these high tunnel systems, we don't have the data that I can say, do a 3 year rotation away from tomato to reduce populations of corky root rot. I just don't have a good answer for that.
Thank you. And we are getting close to our end here. So Matt and Anna, I think we're going to answer these later on, correct?
Yeah, if we want to take one more minute, if there's a quick one, we can
Okay.
address it, but otherwise we should honor people's time and wrap up.
Okay, this seems like a pretty quick question, I think, for Anna. Would sweet sorghum crush stalks work as a carbon source?
Potentially. If you're going to do a cover crop, you want to use a younger stage of the crop because that's going to be easier for the microbes to break down. If you put in a mature sorghum stem, it's likely it would take too long for those microbes to break that down. I do see one other question I want to answer quickly.
Okay.
Is would the anaerobic process produce toxins that can persist? So one thing is the toxins that are produced in ASD, some of them are acetic acid, which is vinegar, and butyric acid, which is the stinky smell in ginkgo seeds. So these are natural products, and they are broken down by other microbes in the soil eventually.
Thank you. Well, we're seeing now that we're kind of wrapping up, and we do want to honor everyone's time and thank you for joining us today. And a big thank you to Matt and Anna for all your expertise and all the panelists that have been helping answer questions. We really appreciate your experience and your time. And this is the last session, but be sure to be looking for follow-up communication by email, including the messages containing information on where you can learn more, and then any answers to the questions that have been posed here today. So again, thank you for participating, and we hope you have a great rest of your day and stay safe.